Good morning. I'm Doug Doerfler, founder of MaxCyte. We talked, the lady before me, the CEO, talked about the solid tumor space, and that's what we're moving into rather aggressively. This is a company you may not know a whole lot about. We've been around for about 20 years. We're listed on the London Stock Exchange. We do a lot of work with many of the companies that are at this great conference, and I want to thank ARM for inviting us to update you all on our progress. So we have a basic technology. It's a non-viral, large-scale clinical technology that we developed specifically for engineering human primary cells. We use this technology in 3 areas. One is in the drug discovery and development space, where we sell the technology to 10 of the top 10 pharmas, 20 of the top 25 pharmas for small molecule discovery, for biologics production, including viral vector manufacturing, that's AAV and lentivirus. We also partner with major cell therapy companies using our technology to engineer cells, and I'll talk a bit about that. We have 70 partnerships now with companies, of which about half of those are licensed for clinical purposes. And we also have our own proprietary program called Pharma. This is a CAR-based program for solid tumors with some very interesting features that I'll talk through with you in the next few minutes. So this is a busy slide, and it's busy because there's a lot of things going on. First of all, what we do is we license our core technology to a number of companies in the field of cellular therapy. And they use our technology. It's a non-viral technology that's clinically oriented to engineer cells. And the block on your left, my right, indicates those research and clinical partners we have relationships with. We have many more. These are companies that gave us permission to use their logo and disclose that they're also working in non-viral cell engineering, which is an area that was discussed earlier today. There's some major advantages in using non-viruses, not using viruses. We'll talk about that with Steve after when I sit down. But you see most of the leaders in the field in terms of cell and gene therapy and in terms of gene editing are using our technology. We've recently announced a number of deals. These are clinical and commercial deals with some of the real leaders in the field. We announced last year a major deal with Precision Biosciences. This is for an allogeneic next-generation CAR-T, non-viral gene editing. The same with CRISPR Therapeutics. We announced that deal last year. This, again, is a clinical and commercial deal for allogeneic-based CAR-Ts, off-the-shelf CAR-Ts. And in the first quarter of this year, we just announced a deal with Kite, a Gilead company. Same basic area. This is off-the-shelf allogeneic non-viral T cells for CARs. So this is an area that we think is massively underserved right now, and really the future of oncology therapies, and we're involved in all of these. At the bottom, what really motivates our company, though, is always staying one step ahead of where the field is heading. We have announced a relationship toward the end of last year with the National Institutes of Health. We're working with John Tisdale at the National Heart, Lung, and Blood Institute. This is a treatment for sickle cell disease. It's a completely non-viral method. You may have seen the report on 60 Minutes on Sunday. That was a virus-based program. This is a non-viral-based program which allows these patients to be multi-dosed during the course of their therapy. So we're very excited about that. And this is actually a single nucleotide swap. So it's taking the A, swap it for a T. We believe that could be a very major movement toward long-term treatment and potentially cure for this disease. Pharma, which is our proprietary program based on our technology, is, we think, very novel. What we're doing here is we're taking peripheral blood monocytes straight from an apheresis instrument. We're then loading messenger RNA into those cells using our technology. This is the flow electroporation technology. And in less than 1 day, we can produce multiple doses for these patients and freeze them down in aliquot. And so the patient would come in once for an apheresis and get anywhere from 5 to 10 or 15 doses for their particular treatment regimen. What's important about this to note is that most other CAR-Ts are in the 5 to 15 day plus manufacturing process. We've been able to get that down to less than 1 day. And what that means is it reduces complexity, it reduces cost, reduces the bricks and mortar. It allows us to think about pushing cell therapy closer to the patient in transfusion medicine centers and away from centralized facilities. And most importantly, it gets treatments to patients faster. And when we talk to oncologists about the current methods of treatment. One of their major concerns right now, concerns of families, is having to wait a month, 6 weeks, 2 months to get a treatment for a disease that we all know is very critical that we treat these diseases as quickly as possible. So let me just stop there and sit down with Steve and talk about what we're doing.
Thanks very much, Doug. So maybe just as an extension to the introduction you just gave, so what the flow electroporation technology, the core technology of the company, how does that work? And how is it differentiated in terms of its features and the resulting cells from viral-based approaches?
So I have a slide on this because I knew these questions were coming, and we're a big believer in visuals. So this is the flow electroporation. We invented this technology. What you see here is a method of using electric forces to drive molecules into cells. Virtually every molecule that's used in engineering has a slight charge on it, and we utilize that charge to move that molecule into the cell. When you think about electroporation, think of a cell and think about a vertical blind. When you walk through the vertical blinds, they close immediately behind you. That's the cell wall. That's basically what we're doing. They're driving the molecule with electric forces into that cell, and we're doing it in very, very large volumes. So we can process 1 × 10⁻⁻⁻ cell, which is a liter of cells, I'm sorry, 100 milliliters of cells, a lot of cells, in about 20 minutes, and 90% efficiency and 90% cell viability. How do we compete against or look at other methods of transfection? I've learned, we're listed on the London Stock Exchange, there's a term over there called different horses for different courses. We're a firm believer that there's a really strong place for AAV and lentivector. We're huge fans of those approaches. We believe that there are going to be important applications for a technology like this that are going to be different than some of the viral approaches. Again, I think you can see by the companies that we've just recently done deals with where we think this is heading. We're seeing a lot of companies moving away from viruses and toward non-viral methods. Most of that reasoning is because a lot of the programs that we see, the companies that have been developed over the last several years, have come out of academia, and companies have really created structures to pull those technologies out of academia. And typically academia is comfortable working in a virus-based system, but when you start thinking about the cost of development, the cost of manufacture, and some basic issues around commercialization, people start thinking about moving away from viruses and toward non-viral systems, including some of the proteins that we're seeing in gene editing and also in messenger RNA, which we're focused. A lot of our work is in messenger RNA.
So, I mean, you highlighted obviously some of the partnerships you have, and you have many of them, and you've been successful, securing those partnerships. So can you just talk a little bit about what that has meant to MaxCyte from the standpoint of revenue and revenue growth? And also, when you do these licensing deals, are you primarily providing IP, instrumentation, training, services, or are these deals structured differently depending on who the partner is?
Yes, everyone's a little bit of a different snowflake, and some of our partners are in the audience today. So every one of these is critically important. One of the longer-term relationships we've had is with Sangamo, with our zinc finger nuclease programs. That has morphed into relationships with BioVertiv and the most recent Kite relationships. So these tend to be long-term relationships. And because they're long-term, there's more than just us supplying an instrument. There's a lot of know-how. We provide a single-use disposable. But again, we try to understand from the customer's perspective what they're trying to achieve. And they're not just trying to put something into a cell. They're actually trying to control a biological function or enhance a biological function. And we have people within MaxCyte, our scientists are just really intently focused on helping develop these medicines. And so there's quite a bit of intellectual property that we can provide to our partners to extend the intellectual property that they have with this delivery method. And we also know that the road in drug development is a winding one. And so we have to be flexible and work with these companies and make sure that we're aligning our interests with their interests. And if they have a change in terms of their strategy, we can work with them to either maybe perhaps when they do a deal with another company, we work with other company to transfer the technology. So we're always there to help them throughout the process, both from a clinical perspective, but also from a commercial perspective.
Great, and just maybe last question on the core technology, and then we can switch to Pharma, I guess. Are there any, I mean, you're testing this with different cargoes and in different indications, so have you come across any restrictions on cell types that you can use it for, or just the scalability? Would this be restricted to orphan diseases, or can it really be scalable to a large degree? And then obviously the cargo, depending on what you're introducing to cells.
Yeah, let's do the cargo first. So first of all, cells don't like DNA put in them. Cells kind of reject that. They don't like that. But there's certain chemistries that we've been able to develop with partners that allow that to happen more readily. In terms of size, we've actually put artificial chromosomes into yaks into cells, which are huge. Again, the cell didn't like it, but we were able to put it in there and activate it inside the cell. A lot of our work is in messenger RNA. It's an area that we think is really capturing a lot of the cell therapy space right now, and directly putting proteins in the cell so that you get around the whole area of having to have the DNA or the RNA make the protein. In terms of volume, this system will do 1 × 10⁻⁻10 cells. We have another system that will do a whole liter of cells, which is 2 × 10⁻11 cells, which is mostly for large-scale biomanufacturing applications, for lenti-vector manufacturing or AAV, or large-scale allogeneic applications. We have several hundred instruments in the field now, so scaling this up and supporting a commercial launch is something that we're focused on. We'll be announcing very soon a new line of instruments that are really directed toward the commercial cell therapy space. Most of the work that we have done, this instrument you see here, is one that really has been developed for the clinical trial work, and now the next step is for us to have a system that can be used commercially, because that's where this is heading. You're seeing a lot of these programs, these clinical programs, although we did talk about small patient numbers, we're seeing FDA really is focused on moving these things forward, these programs forward, because we're working in very serious diseases, and we're seeing really tremendous progress in treating these diseases with cellular therapies. So we're very enthusiastic, obviously, about the space and supporting it.
Great. So I guess let's switch to Pharma now.
Sure.
Introducing mRNA to T cells has implications, obviously, for dosing or redosing, durability, the features of those T cells and how they'll work in folks, and then obviously using it in solid tumors, and in some cases intraperitoneal injection as well versus IV.
Yep.
So maybe just to finish up, could you just describe that program, all those differences that I just mentioned? How does that reflect then in the product, and where you're going to take that program in the clinic?
That's been an hour's worth of discussion.
Well, we have 2 minutes.
2 minutes. So the antigen that we're focused on right now is the mesothelin antigen. We think it's a very interesting antigen from an immuno-oncology perspective. Its frequency and distribution is on a number of malignant cells. We're focused right now, we have a clinical trial that's ongoing. We dosed our first patient in the 4th quarter with the Pharma platform. It's for ovarian cancer and peritoneal mesothelioma. We're proving out safety, and we also have been able to validate our 1-day manufacturing practice. So that's been very important to us, and we'll hopefully see some data coming from that trial toward the end of this year, 2019. We will also we've given guidance, so we'll begin a first in human testing of our intravenous infusion of the same product, mesothelin-directed CARs, and this will be directed toward a broader set of diseases. Mesothelin is found at low degrees on normal tissue, and that's why it's important that we're going with the mRNA route. mRNA is expressed in the cells for a short period of time, then that cell population goes back to its normal state. So we believe we can prospectively manage this on-target, off-tumor toxicity, which has really been an encumbrance for moving CARs into solid tumors. We're currently working with the National Cancer Institute. That's where our first clinical trial site, as well as Washington University of St. Louis, and we'll of course expand that with the IV trial. And I know I didn't answer all your questions, so was there something else in there?
No, I mean, maybe just last, and we only have a couple seconds, but I know there's some phase 1 data from UPenn. There was a trial of mRNA-modified T cells. What did we learn from that, and I guess how does that reflect then on what you'd expect?
Yeah, I think that the previous speaker talked about this a bit, that when you're going after solid tumors, you really do need to reshape the endogenous immune system. And if you can multi-dose patients, which we are doing with this trial, this is a multi-dose trials. So with mRNA, you can do that. With viruses, you tend not to be able to do that. A constant chronic administration of these products. What was shown in the Penn study was there was epitope spreading, and there was a recognition of tumor-associated antigens that were outside of the directed antigens that were on the CAR. So again, the whole ability to recruit the immune system is really important, we think, for treating this next generation of diseases, which is solid tumors.
Great. Fortunately, we're out of time, so thanks very much for the
Steve.
overview.
Thank you.